Hello, my name is Joshua Mugabe. I welcome you to Inheritance. Our lesson objectives today, we are going to look at Mendelian's genetics. We shall look at the mechanism of inheritance, types of zygosity. We shall look at the modes of inheritance. We shall look at the Punnett square. We shall look at the pedigree charts. And we shall look at genetic diseases. You are welcome. Please do not forget to subscribe. Principles of inheritance. Principles of inheritance. Mendel. Gregor Mendel was an Australian monk who developed the principle of inheritance by experimenting on the pea plants. Gregor Mendel was an Australian monk and he developed the principle of inheritance by experimenting on pea plants. He crossed the pure breeding plants with different traits for the same future. For example, he would work on height and he would consider tall or short plant. And they were pure breed. So he crossed the pure breeding plants with different traits. And he could use an example like height. He could cross pure breed tall and pure breed short plants. As a result of these experiments, Mendel discovered that the offsprings produced one trait. The offsprings produced only one of the traits, and they did not blend. The traits were inherited in set ratios. These traits were inherited in set ratio. And when he crossed the F1, he got to the F2, where he got 3 tall and 1 short. So, the offsprings in F1 generation, they only produced one trait. When he crossed the pure breed tall plants and pure breed short plants, in the first generation or first filial generation, he got only one trait, which was tall. But when he crossed to get the F2, now the ratio was 3 were tall and 1 was short. Mendel's experiment. Mendel performed a large number of crosses. He performed a large number of crosses to determine the F2 ratio of 3:1. Let us try to look at some of the experiments that he carried out. He worked on flower color. The dominant trait were the purple, he got 705. Recessive were white, 224, and the ratio was 3.15 to 1. When he worked on the plant height, he had the tall plant, he got 787. The short plants, he got 257, and the ratio was 3.06 to 1. He worked on the seed color. The green, he got 6,022. The yellow, he got 2,001. And the ratio was 3.01 to 1. When he came to the seed shape, he used the round shape and got 5,474. the wrinkled shape, he got 1,850. And the ratio became 2.96 to 1. He looked at the pond color. The green color, he got 428. The yellow color, he got 152. And the ratio came to 2.82 to 1. The pond shape, he had the inflated, he got 882. The flat, he got 299. And the ratio, he got 2.95 to 1. So Mendel carried out large number of crosses to determine the F2 ratio, and his ratio was approximately to 3 to 1. Now, many statisticians, they believe that the results were too precise to actually be genuine. They were too precise to be genuine. They were close. If you look, 3.1, 3.06, 3.01, 2.96, 2.82, 2.95. They are just crossly there. They were not moving very far away. So they were very precise. So for them, they say that they were precise to actually be genuine. Mendel's conclusion and current day understanding. 1. The organisms have inheritable factors. That is what he included. Current understanding, these factors are now the ones that we call the genes. During Mendel's conclusion, he said that there are a version of each factor. Today, genes have alternative alleles. Then he also concluded that parents pass on only one version. And current understanding, gametes or the sexy cells are haploid. He also concluded that parents contribute equally to inheritance. Today we know that offspring body cells are diploid. And lastly, he concluded by saying that only one version of the factor is expressed. And here we say dominant versus recessive allele. Mendel's law. Mendel's observation led to the establishment of a certain fundamental rules. His observation led to the establishment of certain fundamental rules. 1. The law of segregation. Here it says that alleles are separated. The alleles are separated when haploid gametes form. We saw that one in meiosis. Segregation. Alleles which are on different chromosomes are separated when haploid gametes are formed. Secondly, the law of independent assortment. Here, separation of allele for one gene occur independently of the separation of allele of other given genes. This one we saw it in also metaphase. The independent assortment, separation of alleles of one gene is independent of the separation of alleles of any other given genes. That is when there is alignment of homologous chromosomes on the spindle. When the homologous chromosomes align themselves on the spindle, the separation of those chromosomes is independent of the separation of other chromosomes of given other gene. So when we go to anaphase 2, anaphase 2, now we are separating sister chromatids. And these sister chromatids, when we are separating them, they are independent also of the other chromatids of other chromosomes. Then the principle of dominance. Recessive alleles will be masked by dominant alleles. Here you find that a dominant character is going to mask a recessive character, or a gene that is dominant is going to mask another gene which is recessive, which is the allele for a dominant character is going to mask the allele of a recessive character. But certain caveats to these rules are known to exist. There are 7 caveats to these rules which are existing. Genes on the same chromosomes do not independently assort. That means that the genes are linked. When genes are on the same chromosomes, they don't independently assort. They all go together in one direction. That is 1. 2, not all traits show complete dominance. Not all traits show complete dominance, because there are sometimes when we have codominance and partial dominance. So it's not that a dominant will always mask the recessive. Not all traits show complete dominance. That is a caveat. Let's look at the principles of inheritance. We are looking at the genes versus alleles. Inherited features are encoded by a specific DNA sequence called a gene. Inherited features, or what we can call characters, those ones are encoded by a specific DNA sequence. And that DNA sequence is what we call the gene. Variation of the specific feature are encoded by allele, or what we call alternative form of genes. So we are saying that a specific feature is encoded by a DNA sequence, which we refer to as a gene. But the variation in that specific feature, variation in that character, is encoded by alleles, or the alternative forms of the gene. Let us try to look here. We have our chromosome here, and this chromosome has a gene up here, is a gene for eye color. Then we have another gene down here, is a gene for hair color. That is a gene which encodes the specific feature, which is the eye color and hair color. But again, when you look at the eye color, there are those eyes that are brown, there are eyes that are blue, there are eyes that are green. Now that one, it is the same gene, eye color, but it is variation. So now that variation of having eye color, brown eye color, blue eye color, green eye color, that one now is encoded by alleles. Alleles, these are alternative form of that particular gene. A brown eye color, it is a gene. A blue eye color, it is an allele, which is the same gene. A green eye color is an allele, which is the same gene of eye color. Now, when you come to hair color, you have the broad, you have the black, you have the ginger. All those ones are hair color. It is one gene. But the broad, the black, the ginger, those are alternative form of a gene, which we call the allele or the potential alleles. Diploid versus haploid. Genes are located on a specific position on a chromosome, and that position is known as a gene locus. So when you are looking for the genes, you will find them at a specific location on the chromosome, and that specific location is what we call the locus. For example, gene A is located at this position of the chromosome, which we call the P position. Gene B is located at this position, which we call the Q position. So they are located at specific position on the chromosome, and those positions are the ones that we call the locus. So this is a locus for gene A, and this one is a locus for gene B on a chromosome. Every gene has a specific location. Every gene has a specific location on a particular chromosome. On a particular chromosome. For example, chromosome 7. Then we have location 2, and then the binding position 31.2. That is a location of a particular gene. You can find it at that particular location. Chromosome number 7 on Q and at binding position 31.2. Offsprings inherit chromosomes from both parents and so have chromosome pairs. So here we are seeing that an offspring is going to inherit the chromosomes from both the parents, which means that each parent is contributing half the number of chromosomes. Haploid, when they come together, they form a diploid. Only one chromosome of each homologous pair gets passed to the offspring. It's only one chromosome of each homologous pair gets passed to the offspring. Here we are looking at the gamete. Each gamete is having haploid number of chromosomes, which means that, for example, if we are considering human species, Homo sapien, they have a total of 46 chromosomes. Those are 23 pairs. So of those 23 pairs, or the 46 chromosomes, only 23 chromosomes will be passed to the offspring. So each gamete will be having half the number of chromosomes. So the paternal will bring 23 and the maternal will bring 23, and when they combine, now they form the 46. So the parent, each parent brings only one chromosome of each homologous pair. That is what he or she passes to the offspring. Consequently, body cells are diploid, that is with 2 copies of each chromosome. they have 2 alleles per gene. So here, when we are talking about the body cells, for the body cells, they have diploid number, meaning that they have 46 chromosomes, which makes up 23 pairs. So each pair of a chromosome, one is from the maternal and another one is from the paternal. So which means that every gene which is on the particular chromosome has 2 alleles. So there are 2 alleles per gene. 2 alleles per gene in a diploid body cell. Sexy cells are haploid with only 1 copy of each chromosome. The sexy cells, for them, they have half the number of chromosomes. That is 1 copy for each of the chromosome. 1 allele per gene, that is in the sexy cell, or what we call the gamete cell. They have only 1 chromosome, 1 copy of a chromosome of the homologous pair. And that 1 copy has got only 1 gene or 1 allele. Each gene has got only 1 allele. Allele segregation in gametes. Allele segregation in gametes. The formation of haploid gametes is achieved by the process of meiosis and subsequently gametogenesis. So if we are to form a haploid gamete, the cell undergo division under the process called meiosis. In meiosis, we end up halving the number of chromosomes, changing the chromosome number from being diploid to being haploid. And this one occurs in the formation of gametes, the sperms and the ovum or the eggs. So we end up with the cells which are haploid. Haploid means that they have half the number of chromosomes. Let us look at our cell here. We have our cell here. It starts with a homologous pair of chromosomes. Now, when we follow that homologous pair of chromosomes, here it condenses and then we can see them as homologous chromosomes. The other side, they were chromatid. Now, during meiosis, we separate these homologous chromosomes, and each of the chromosome goes in its own cell. Now, that one is what we are saying. Now they become haploid. Haploid, each cell has got half the number of the total number of chromosomes that has supposed to be there. Then here in the second meiotic division, it is mitotic division. We are just separating the sister chromatids, and each sister chromatid goes into its own cell. And therefore we form 4 daughter cells which are haploid, having half the number of the original cell chromosome number. Meiosis separates the chromosome pairs, or what we call the allele pairs, into separate gametes via a reduction meiosis. So during meiosis, we are going to separate the chromosome pairs. Here we have our pair of chromosome that we have already seen here. This is one pair of the chromosome. So meiosis, what it is going to do is going to separate this homologous chromosome or this chromosome pair, and now each chromosome will go in its own cell. That is what we are referring to as meiosis. Separates the chromosome pair or allele pairs into separate gametes. So this one is called a reduction division. Reduction division. Segregation of alleles is random. So inheritance patterns of different genes are independent of another. So here what we are saying, that the segregation of the alleles is random. It just happens anyhow. And so the inheritance pattern of different genes are independent. If, let us say, we had another chromosome here, and then also another chromosome here, they are in the same, we can say they are in the same cell. If they are the same cell, what we are saying that allele segregation is random. The segregation of this allele and this one is going to be random. It is not going to be say that this one, this color and this color will go this side, and then we say the red color will go the other side. No, it is not like that. This side we can have red going with blue, and the other side is still we can have blue going with red. Or it can be that only this side we can have red and red, then the other side we can have blue and blue. So it is independent. It is independent. The inheritance pattern of different genes is independent of another. Where this one goes, it does not matter where the other one is going to go. So unless the genes are on the same chromosome, that is leakage. Unless these genes are on the same chromosome. Remember up we have said that the alleles segregation is random. Where the allele which is on one chromosome, for example, the allele here and the allele here, their separation is independent. They can go each one its own ways. Unless those genes are on the same chromosome. If those genes are on the same chromosome, therefore they are not independent of one another. All of them, if this one goes this side, all of them, they will go on the same side. So the same applies. The allele that we have here, or the gene, and another gene which we have here, they are not independent of each other because they are linked. They are linked on the same chromosome. Wherever this one goes, also the other one, that's where it will go. So that's what we are saying, that these ones, the allele here, they are not independent because they are linked. They are on the same chromosome. Therefore, they are not independent of one another. But if the alleles are on different chromosomes, this one here and this one here, they are on different chromosomes, therefore they are independent. Their inheritance character or pattern is going to be different from another gene's movement. The two alleles of each gene separate into different haploid daughter nuclei during meiosis. Here, let's now go back here. We have this chromosome here. Let's look at this homologous chromosome. On that homologous chromosome, we have an allele here. And then on the other one also, we have an allele here on those two homologous chromosomes. Now we are saying the two alleles of each gene, we can say that they are the same gene. The two alleles of each gene, separates into different haploid daughter nuclei. So this one is going to go this side, and the other one will go the other side. So they have separated and they have gone into different haploid daughter nuclei. What do they mean, haploid? Here they were diploid. They were two in the same cell. Now here each one is independent. One. That is now what we are calling haploid daughter nuclei. So they are going to separate. They have been two alleles of the same gene, but now they are separated into different haploid daughter nuclei, and this one occurs during meiosis. So here it is meiosis. Let's go to sexual reproduction. During sexual reproduction, the fusion of gametes results into a diploid zygote. During sexual reproduction, fusion of these gametes is going to result into a diploid zygote. Now, this is what we are saying, that if we have a man and this man produces gametes, this chromosome here is going to go in this sperm, and this chromosome here will also go into another sperm here. So which means at first they were together and they were diploid too. But now they have separated. Now they are haploid. Now the same applies to Our Lady. She has this chromosome here. This chromosome is going to separate and it will go in this egg. And then also there is this chromosome here. This chromosome also here is going to separate and it will go in this egg. So now you can see that every egg has got one chromosome of the pair. And that one is what we call haploid. So what we are meaning is that this sperm here is haploid. It has one chromosome of the pair. And this one is haploid, one chromosome. The same applies to this egg. This egg is haploid. Even this egg here is also haploid. But when we go to the fusion of those gametes, let us say that this sperm here is going to fuse with this egg. When they fuse together, this one is going to bring a chromosome, and this one will bring also a chromosome. Now those two chromosomes will be a pair, and that is what we call now a diploid zygote with two pairs of the same chromosome. That is what we are referring to as a diploid zygote. The zygote becomes a diploid. So during sexual reproduction, the fusion of gametes is going to result in a diploid zygote. Now, we are saying with two copies of every chromosomes, there are two alleles of each gene. I want us to understand this one. Two copies of every chromosome, there are two alleles for each gene. Now, let's look here. Here we have a chromosome, and this one is a chromosome. Those two chromosomes, they form a pair which we call a homologous chromosome. And at a particular point on this chromosome, let us say at this point on that chromosome, there is a gene. And also at that same particular point on the other chromosome, the other homologous chromosome there is also a gene. The gene here is small a. The gene here is capital A. It is the same gene. The other one is small a, the other one is capital A. Now, we call these two alleles. Those two are called alleles. They are the same, it is the same gene, but different alleles, alternative form of that same gene. So we are saying that with 2 copies of every chromosome, this is the same chromosome, there are 2 alleles. There are 2 alleles for each gene. Okay? Now, this is the same chromosome as this one. They are what we are calling the 2 copies. It is the same chromosome. They are the same chromosome. This is chromosome is the same as this chromosome. And this is a gene. And this one is also a gene. They are located at the same point or the same locus on the different gene. Those 2 genes are now what we are calling the alleles. And they are of the same gene, but different alleles. Allele combination, the allele combination that is inherited is determined by random probability. The allele combination that is inherited is determined by random probability. So what is going to be inherited is going to be random. For example, let us look here. When you look at this sperm here, it has allele capital A. When you look at this egg here, it has allele capital A. So when these 2 combine, we shall have capital A, capital A. That is allele combination that has been inherited. But again, this one here may now not go with the other one. It may end up going with this one. So now the allele combination will be capital A, small a. That type of allele combination that is inherited is determined by random probability. There is a probability that this one will combine with this one, or it will combine with the other one. It is a probability. It is not a guarantee. It is a probability. The same we can look at this one here. If you look at this sperm has a probability of going with the other one, and they will form capital A, small a. And it also has a probability that instead of going with the other one, it can go with this one. And they will have a combination, a little combination of small a, small a. So, which one will it go with? It is just random. It can go with the either of any of those other allele. The fusion of gametes results in a diploid zygote with 2 alleles of each gene. So when we bring this sperm here and it fuses with this egg here, what they are going to give us is going to be what we are calling our zygote. We are going to receive our zygote. And the zygote that we are getting is having 2 alleles. It has allele A, capital A, and allele capital A. And those 2 alleles, when they come together, now they are the ones that we are calling the diploid. So our zygote is going to be diploid with the two alleles, capital A and capital A, of each gene. This is allele A of the same gene, and this is allele A of the same gene. Types of zygosity. Types of zygosity. There are three types of allele combination. We have three types of allele combination. And which ones are these? We have the homozygous. Homozygous type of combination is whereby the alleles are the same. So if you look here, this one that we have here is a homozygous type of zygosity. Both alleles are the same. If you look at the alleles are the same. Then we have the heterozygous. Heterozygous, this is where alleles are different. The alleles are different. Now here, if you look at these two, the alleles are different. We have capital B and small b. That's what we call the heterozygous. The zygosity is heterozygous. Alleles are different. Then the third one, we have the hemizygous. Here, there is only one allele. We have only one allele. For example, if you look at this one, there is only one allele, which is the C allele. There are two, but there is only one allele. The other one does not have any other allele. So we refer to that zygosity as hemizygous. Hemizygous combination occurs in males as their sex chromosomes are different. This hemizygous combination, it occurs in male as their sex chromosomes are different. So when you come to hemizygous here, you know that we have the X chromosome and then the Y chromosome. The Y chromosome is the one which is short, and the X chromosome is the one which is long. So there are some of the alleles which are on the X chromosome, but they are not on the Y chromosome, because the Y chromosome is a short chromosome. So it cannot have all the alleles which are on the X chromosome cannot be fixed on the Y chromosome. So that's why it forms the hemizygous, and it occurs only in males as their sex chromosomes are different. The males, for them, they have X and Y chromosome. The two alleles of each gene may be the same gene, or they may be different alleles. So we are having, this is the same gene here. That is the same gene, that is the same gene. This one is the same gene and this one is the same gene. They are located on homologous chromosome and at the same locus. So it is the same gene. So you find that now we refer to them alleles. They are alleles. Now, if you look at this allele here, this allele is the same as this allele, capital A, capital A. But again, this allele here is different from this allele. So these ones is what we are saying, that they are the same genes, but they may be the same allele or they may be different alleles. So the two alleles of each gene, that one is the same gene, this one is the same gene, but these ones are the same, these are the same, these ones are different alleles. Please don't forget to subscribe. Genotype versus phenotype. We want now to look at the genotype versus the phenotype. Allele combinations for any given characteristics is called a genotype. The allele combination for any given characteristic is called a genotype. For example, let us look here. Here we have our homologous chromosome. They have come together. But we know that those homologous chromosomes, this one might have come from the paternal and this one might have come from the maternal. But they have at the same locus there is a gene. And the same locus here on the maternal, there is a gene. That one is the same gene. And because they are of the same gene on different chromosomes, therefore we refer to them as alleles. So these two are homozygous alleles. Now this combination of this allele of a given characteristic, now the characteristic here we are talking about is the hair type. This characteristic. Now this one is what we call the genotype. BB. BB is our genotype. It is the allele combination of a given character, is what we refer to as a genotype. BB is a genotype. Now, a genotype may be homozygous, that is when they are all the same. Like for example, here it is homozygous. It might be heterozygous. For example, if one of them is capital and another one is small. Or they can be hemizygous, like the one that we have seen in sex chromosome of males, whereby we find it on only one chromosome, the X chromosome, the allele, and it is not existing on the Y chromosome. Phenotype. Phenotypes are influenced by the genotype and the environment. A phenotype is influenced by the genotype and the environment. In the real sense, a phenotype is an expression of a genotype. A genotype expresses itself into a phenotype. For example, here we are having a genotype of hair color. A genotype of hair color. This one is on the chromosome. So it expresses itself by giving the hair a color. Now it has given a blonde hair color. It has given a blonde hair color. The color which is seen is what we call the phenotype. And the genes that we cannot see that are on the chromosome are what we call the genotype. So a phenotype is going to be influenced by the genotype and then also by the environment. Patterns of inheritance. Patterns of inheritance. Now, modes of inheritance. Let's look at the modes of inheritance. We have a monohybrid cross. We have a monohybrid cross, and this one involves mating with reference to a single trait of interest. Here we are looking at this mode of inheritance, which is monohybrid cross. It involves mating with reference to a single trait of interest. We are looking only at the trait that we are interested in, and we are going to cross only that trait. For example, the trait might be height. We are interested only in height, not in color, but height. So if we are interested in height, we are going to mate in reference to only that single trait, which is height. So we can mate a tall crossing with a short one, or a tall crossing with a tall one, or a short one crossing with a short one. We are only looking at one character at a time, only a single character of interest. It is what we call monohybrid cross. The outcome of the monohybrid cross can be calculated via a Punnett grid. We can calculate what is going to come out of this cross using a Punnett grid, or sometimes we call it a Punnett square. The pattern established will be governed by the mode of inheritance. The pattern established is going to be governed by the mode of inheritance. For example, we have what we are calling complete dominance. In complete dominance, it's where one allele is expressed over the other. One allele is expressed over the other, meaning that the other allele is going to be masked. So what we are going to see in our phenotype is going to be only the expression of one allele. The other allele is going to be masked. Let me give a simple example. If, for example, we were looking at height, crossing a tall plant with a short plant. When we cross those two, we expect the two tall and short to express themselves in the offspring that we are going to have. But we may find that only one allele is expressing itself, and we find out that offsprings that we get after the crossing, they are all tall, which means that the allele for tallness is the one which is expressing itself in the phenotype, in the offspring. So now that one means that the allele for tallness is dominant to the allele for shortness. So that one now, because it is expressing itself and it is being seen in the phenotype, and the shortness allele has been suppressed or masked, it is not expressing itself in the phenotype, therefore we say that one is what we refer to as complete dominance. Then we have codominance. Codominance here, both alleles are going to express themselves equally in the phenotype. So here we are having both alleles expressing themselves. If I cross a brown hen and then I cross it with a white hen, and when they produce the chicks will have both the color brown and white specks, meaning that both colors are expressing themselves. Therefore, that is what we are calling codominance. Both of them are expressing themselves. Both alleles are expressing themselves in the phenotype. That is what we call codominance. Sex linkage. We have sex linkage. Now, in sex linkage, alleles are located on the sex chromosome. The alleles are located on the sex chromosome. The trait shows sex bias. The trait is going to show sex bias. Let us say we have X chromosome and Y chromosome, and this allele is on the X chromosome, but it is not on the Y chromosome. So when it is going to express itself in the phenotype, it is going to show sex bias. It may express itself in the boys, and it may fail to express itself in the females. So that one shows that it is sex linked, or what we refer to as sex linkage. The ratio established represents the probabilities and may not mirror the actual results. The ratios that we will establish will represent the probabilities and may not mirror the actual results. Let's look at the Punnett grid. The outcome of a monohybrid cross can be represented using a Punnett grid. We can represent it using a Punnett grid. This designates characters to represent alleles. For example, A represents dark red, and small a represents light red. Now we have the male this side, and these are the alleles for the male. A representing the dark red and small a representing the light red. Then this side we have the female. These are the alleles. Capital A representing the dark red and small a representing the light red. So let's see how we cross. First of all, you write the genotypes and the phenotype of the parents. So the parent here is a female. That is the phenotype. And here is a male phenotype. The genotype, we have capital A and small a. Capital A and small a. Those are our genotypes. Use the grid to work out the gamete combination. See step shown below. So now we are going to use the grid to work out the gamete combination. We already have capital A here, small a here, capital A here, and small a here. Here now we start with small a. If you look at our small a here, it moves from here and it goes here, and then this small a here, when they come here, they form small a, small a. That one we have finished. Now we go again on small a. Here we are going to cross this small a with this capital A. Now when they come, they will form capital A, small a. Now we have finished crossing small a here. Now we go to our big A here. Now this big A, we are going to cross it with the small a. When you cross, we shall get capital A and small a. Now after crossing it with that one, now we shall come and cross this big A with this capital A. So when you cross them, this one will bring and this one brings, and then we shall get capital A, capital A. So now our Punnett grid is filled. We have capital A, capital A, capital A, small a, capital A, small a, and small a, small a. So those are our gamete combination that we have got, and we have used the grid to work out our gamete combination. So write the genotype and the phenotype ratio of the offspring. You will see that we have 3 dark red and 1 light red. Let us look at this one. So when you look here, you are going to see that we have capital A, small a, that one is going to be dark red. Capital A is going to be dark red. Capital A, small a is also going to be dark red. And then we have small a is the one which is going to be light red. So you will see we have 3 capital dark red and we have 1 light red. Complete dominancy. Complete dominancy. Now, complete dominancy is an expression of one allele over another. And this one is seen in heterozygotes. Here in complete dominancy, it's whereby one allele is expressing itself over another. And it occurs in heterozygotes. For example, let us look here. We have the phenotype. Phenotype is an outside expression. Our phenotype, we have a black rat, we have a black rat, and we have a brown rat. That is the phenotype. Then we have the genotype. We have the genotype. The genotype, we have capital B, we have capital B, small b, and we have small b. So of all those ones, which one is a heterozygote? Which one is a heterozygote? A heterozygote is that genotype which have the recessive, the dominant allele and the recessive allele. We call it a heterozygote. They are different alleles. Then the other ones which have got the same alleles, those ones we call them homozygotes. Those are homozygotes. These are the same, capital B, homozygous dominant. Then this one, they are the same, capital B, small b, they are the same, they are homozygous recessive. So we are saying that complete dominancy is expression of one allele over another. When we have a dominant allele, which is capital, written like capital, and we have another one which is a recessive allele, which is written as a small case letter. That means that the one which is going to express itself is the one that we have written in capital letter. It becomes the dominant. So if capital B is representing for black and small b is representing for brown, the one which is going to express itself is the one which is dominant, and capital B is dominant. Therefore, this rat which has got two alleles, one is dominant, another one is allele, the dominant allele is going to express itself. And here the dominant allele is representing black, and the recessive allele is representing brown. Therefore, the color of the rat is going to be black. So, dominance, complete dominancy. This is an expression of one allele over another. And this one occurs in heterozygotes. This is our heterozygote combination. We have one allele, which is capital B dominant for black, suppressing or expressing itself over the recessive B, which is the small b, in the heterozygote. The allele that is expressed is dominant. The allele that is expressed is dominant allele, and it is represented by capital letter. We have already seen that one. This one is dominant and is expressed in capital letter. So it is the allele which is going to express itself. The allele that is not expressed, is recessive. The allele that is not expressed is recessive, and it is represented by a lowercase letter. And you can see here our recessive allele is expressed as a lowercase letter. Therefore, that one represents for brown color. Therefore, it will not express itself in a phenotype because it is recessive. Let's now look at codominancy. Codominancy is when both alleles are expressed equally in a heterozygous. Codominancy is when both alleles are expressed equally in the heterozygote. For example, here we have the expression here, the color. The color is going to be expressed in codominancy. We have capital B, color C capital B, that is for black. C white, it is going to give us white. But when it comes to C black and C white, now both of them are going to express themselves. And that is what we are calling the speckled. So when they express both of them, the combination is a heterozygote. Black and white, they are different alleles or alternative allele of the same gene, which is color. But now both of them are expressing themselves. And if they are expressing both of themselves, it means that there is no one which is dominant and there is no one which is recessive. All of them are codominant, and therefore they express themselves in the phenotype. That's why you see the hen is not only black, but it also has white speckles. Superscript annotations are used to represent the different codominant allele. We are using superscript notations that are used to represent the different codominant alleles. For example, here we are using capital B. Those are superscript, and they are the ones that are representing the codominant allele. Here we have capital B, capital W. So we use them as superscript so that they can represent the different codominant allele. And the main character color C is the color. Dominant alleles mask the effect of recessive alleles, but codominant alleles have a joint effect. If we are talking about the dominant alleles are those ones that mask the recessive alleles. But codominant allele have joint effect. So if we say that it is a dominant allele, that means that one is dominant over the recessive. So the dominant is going to mask the effect of the recessive. But when we say that they are codominant allele, all of them, they have a joint effect. All of them, they will express themselves. Codominance in ABO blood groups. Codominance in ABO blood groups. ABO blood genes, ABO genes have multiple alleles. They have multiple alleles, and those are 3, and it demonstrates codominance. They demonstrate codominance. Now we are in blood groups. Blood groups, they have ABO genes, and these ones are multiple alleles. They are 3. We have seen that the other characters are being controlled by 2 alleles. But now when it comes to blood group, we are seeing that it is being controlled by 3 alleles. Therefore, we call it multiple alleles. And they express or demonstrate codominance. For example, we are using superscript to represent the allele. Allele A and allele B, both are codominant allele, while I is a recessive. Now that one would be allele O is a recessive. So immunoglobulin A and immunoglobulin B, those ones show codominance. All of them, they express themselves where they are existing together. It is only O which is recessive. Now when we go down here, we can see that we have blood group A. Blood group A has antigen A. It has got antigen A on its surface. You can see it has antigen A on its surface, which is identifying it. Now, when you look at the genotypes, we have A and A. It can be A and A, or it can be A and O. O is a recessive. So when O is with A, it is going to be masked or suppressed. So it will be now blood group A. When you go to blood group B, that is the blood group type. And when you go to the genotype, it has allele B, capital B, and allele capital B. It can be the genotype. And if you look at another combination, it can be allele capital B and allele O. Those are the possible genotypes of blood group A. The red blood cell has got antigens on its surface. It has antigen B. It has antigen B on its surface. It has got antigen B on its surface. So antigen B is its surface or identifier. And when you go to blood group AB, when you go to blood group AB, the genotype, it has got allele A and allele B. And when you look at its surface, it has both the genotypes. It has both antigens. Antigen A and also has antigen. It has got antigen A. It has antigen A, and it has also got antigen B. So on its surface, it has both antigen A and antigen B. Therefore, that one is the phenotype, blood group AB. Then when we go to blood group O, if you look at blood group O has I and I, O and O, and for it does not have any antigen, no antigen at all, and therefore it is blood group O. So when it is capital A and A, when it is A and A, there it is only A. A and O, A suppresses O. When it is B, B and B, they are the same. B and O is masked. When it comes to A and B, both of them, they are codominant alleles and they express themselves. And therefore the phenotype becomes A and B. They express themselves, both of them. Sex linkage. Sex linkage, this one refers to when a gene is found on the sex chromosome. Sex linkage is found, or it refers to when a gene is found on the sex chromosome. It can be on the X chromosome, it can be on the Y chromosome. Now, if you look at our X chromosome, you are going to see that the X chromosome has got 153 million base pairs. The bases that you find on this chromosome, those bases that you find on the chromosome, they are 153 million base pairs. Those are the bases that you're going to find on the X chromosome, which is approximately 2,000 genes. So this chromosome can be having approximately 2,000 genes. Now, when you go to the Y chromosome, if we go to the Y chromosome, we shall find here that for it, it has 50 million base pairs. The bases that we are going to find on it are only 50 million base pair compared to the X chromosome, which has got 153 million base pairs. Now, when you look at the number of genes that we can find on the Y chromosome, they are 78 genes. We can only find there 78 genes. Therefore, if they are homologous, the X and Y are homologous chromosome, it means that chromosome X has 2,000 genes and chromosome Y has 78 genes, which means that chromosome X has more genes compared to chromosome Y. Therefore, there are some chromosomes that will find on the X chromosome which you will not find on the Y chromosome. Therefore, those chromosomes that we find on the X chromosome and they are not on the Y chromosome, we refer to them as sex-linked genes. They are sex-linked genes. Sex linkage refers to when genes are found on a sex chromosome. Sex-linked conditions are usually X-linked. Sex-linked conditions are usually X-linked, as very few genes exists on the Y. So most of the conditions that we have, and we call them sex-linked conditions, they are usually on the X-linked, or they are X-linked. They are found on the sex X chromosome because there are few genes on the Y chromosome. So, sex-linked traits exhibits different patterns of inheritance. Sex-linked traits, they are going to exhibit different patterns of inheritance. This is because, 1, females cannot express Y-linked traits. Females cannot express Y-linked traits because they lack the Y chromosomes. So the traits that are found on the Y chromosome cannot be expressed by females because these traits are only found linked on the Y chromosome, which the females cannot have. Males cannot mask excess linked recessive traits. Males cannot mask excess X-linked recessive trait. They cannot suppress X-linked recessive traits because the males, they are hemizygous. Therefore, they cannot be carrier. Let us give an example. If the hemophilia condition is on the X chromosome, that hemophilia, when it appears in the males, it cannot be masked because the males are hemizygous. So males will be affected either by color blindness, hemophilia, because they cannot be carrier. They cannot mask that X-linked recessive trait which is on the X chromosome. The pattern of inheritance is different with the sex-linked genes. This is due to their location on the sex chromosome. The pattern of inheritance is different with the sex-linked genes due to their location on the sex chromosome. As we have seen that we have the X chromosome and we have the Y chromosome. So we are saying that the pattern of their inheritance is going to be different with the sex-linked genes because of their location on the sex chromosome. Let us say now we are looking at this gene which is located here. If that gene is located here, when you come to the Y chromosome, there is no other gene which is going to be located there. So the type of inheritance is going to be, the pattern of inheritance is going to be different for that gene. Compared to this gene here, if you look at this gene here, there is a corresponding gene here on the Y chromosome. So these two genes, their inheritance is going to be different because of their location on the sex chromosomes. Please do not forget to subscribe. Sex-linked inheritance. Let's look at sex-linked inheritance. For X-linked dominant, that is XR, sex-linked dominant. For example, if you look here, we have the mother. The X is recessive. And when you look at the father, the X is dominant. So when the crosses are taken, you can see that the X of the father goes to the daughters. It goes to the daughters. The X dominant, and the other ones that are recessives will go to the son. These ones are coming from the mother. So, affected father will also result in affected daughters. So the father is affected, and then the daughters are also going to be affected, because the X, which is linked with the trait that is dominant, that X goes to the daughters. It cannot go to the sons, so it goes to the daughters. So if the father is affected, then the daughters are also going to be affected. Now, for X-linked recessive, if we look at the X-linked recessive. Now the X-linked recessive, you can see that it is with the father. Now this X-linked recessive, which is the father, is going to go with the daughters. It will go with the daughters. And X, of course, the daughters will also get the X-linked dominant from the mother. Now, when you go to the sons, they get the X-linked dominant from the mother. They are getting the X-linked dominant from the mother, because it is coming now from the mother. So what is going to happen? The mother is going to be affected, and as the mother is affected, therefore also the sons are going to be affected. So you can see that the X has been passed over to the son, and the son does not have the X-linked recessive to suppress the X-linked dominant. So the son is also going to be affected like the mother. Pedigree charts. A pedigree chart maps the genetic history of a family over several generations. What we call a pedigree chart is that one that shows the genetic history of a family. So the generations are going to be labeled by Roman numbers. This is generation 1, generation 2, generation 3. Those ones that we are seeing there, the generation. The first generation, the second generation, and then the third generation. So those Roman numbers are representing the individuals, and they numbered by age. Individuals are numbered by age. The individuals are numbered by age. So we have 1, 2, 3, 4. Those ones that are numbered, they are individuals numbered by age. Those ones, this one, they are numbered according to their age. Men are represented by squares. Females are represented by circles. So where you see a square, that is a male. Where you see a circle, that is a female. And the shaded represent a trait. That person has got a trait. Where you see the shade, that individual has got a trait. So here we can see the trait is with the father, and when the trait is with the father, it is not passed over to the children. The trait is not passed over to the children. And here we are seeing the trait is with the mother, and it goes on and it is passed over to her son, one of her son, but other son is free. So if the son also marries a woman with also the trait, now you will see all their children are having the trait. Pedigree chart, still dominant versus recessive. For autosome dominant traits, these are autosomal dominant trait. The father has a dominant trait, and if also the mother has a dominant trait, so we expect the boy to have a dominant trait and then the girls to have a dominant trait. Now there is this one here without the trait, but they are saying that this one could not occur if the trait was recessive. Here there is one girl who does not have the trait, but they are saying the trait could not occur if the trait was recessive. If both parents are affected and any of the offspring isn't, then the trait is dominant. That trait is dominant. We have both the parents are affected, but one of the offspring is not. Then that means that trait is dominant. The trait might be dominant. Then we go for autosomal recessive trait. Autosomal recessive trait. Now here we are seeing the father does not have the trait, the mother does not have the trait. And when you come here, some of the children don't have the trait. But again you find that one of the girls has the trait. And here we are saying it could not occur if the trait was dominant. This one could not occur if the trait was dominant. Now the trait means that the trait is expressing itself in a recessive manner. So where we have the recessive, therefore the trait is going to express itself. So, but if the trait it was dominant, then it would not occur. So here we are saying if neither parent is affected and any of the offspring is, then the trait is recessive. The trait is expressing itself in the recessive form. We go to genetic diseases. Genetic diseases. Genetic disorder is caused by altered genes or sets of genes. The genetic disorder is caused by altered genes. Altered genes, these are genes that have been changed. So genetic disorders, these ones include a single gene disorder. It can be chromosome abnormalities. It can be mitochondrial diseases or multifactorial or polygenic disorders. Those are genetic disorders. They include the single gene disorders. They can be chromosome abnormalities, mitochondrial diseases, or multifactorial or polygenic disorders. Many genetic diseases have been identified in humans, but most of them are rare. Most of them are very rare. So let's look at single gene disorder. In single gene disorder, this one occurs when a mutation to a single gene alters or abrogates the protein function. And this one leads to the development of a disease phenotype. Here we are having a single gene disorder which has occurred as a result of mutation. And this one, there is a single gene alter which is going to cause the abrogation of the protein function, and it is going to lead to the development of the disease phenotype. For example, of a single gene disorders, this one includes autosomal dominant, like for example, the Huntington disease. We have autosomal recessive. Here we have cystic fibrosis. We have autosomal codominant, where we have sickle cell anemia. All those are examples of single gene disorder. And then there is X-linked recessive. X-linked recessive, where we have hemophilia and red-green color blindness. Those are X-linked recessive. Gene diseases, genetic diseases, we start with the Huntington disease. The Huntington disease is an autosomal dominant disorder disease. And what happens? It is caused by mutation. It is caused by mutation to the HTT gene, which is on chromosome number 4. The HTT gene contains a repeating sequence of CAG, cytosine, adenine, and guanine. Normally, there are low number of repeats. There are a low number of repeats, which are less than 25. That is normally. But more repeats, which are more than 40, these ones are going to cause protein to misfold. When the repeats are many, for example, the sequence of repeating the particular bases is more than 40, it will cause the protein to misfold. And of course, a misfolded protein will cause what we call neurodegeneration. When the protein is misfolded because of over-repetition, it is going to cause neurodegeneration. This often occurs in late adulthood. It often occurs in late adulthood, and it leads to dementia. It leads to dementia. Then we have cystic fibrosis, still a genetic disease. In cystic fibrosis is an autosomal recessive disorder. It's an autosomal recessive disorder, and it is caused by mutation to the CFTR gene, which is on chromosome number 7. It is a mutation. Individuals with this disorder, they produce thick mucus, and this mucus clogs the airways and the digestive tract, and this can lead to respiratory failure and pancreatic cystic. So this, if you look here, we have the mucus which is produced. A lot of mucus is produced in the airways. And that mucus is going to block the airways or the digestive tract. And when it blocks the digestive tracts or the airway, then we are going to have respiratory failure. Or we shall have what we are calling pancreatic cysts. Then, heterozygous carriers do not display disease symptoms. Those who are heterozygous, who are carriers, they don't display the disease symptom. This is because the presence of a normal protein prevents the excess mucus. And because they are heterozygous carriers, they have the abnormal protein which has misfolded, and then they have the normal protein. The normal protein is going to prevent the excess secretion of mucus. Please do not forget to subscribe. We are still on genetic diseases. We have the red-green color blindness. Now, the red-green color blindness is an X-linked recessive condition. Red-green color blindness is an X-linked recessive condition. Therefore, it is more commonly found in males who are hemizygous, because males cannot be carriers because they have the X and Y chromosome, and yet the Y chromosome is not the same as the X chromosome, so they cannot be carriers because the trait is carried on the X chromosome. So the condition is caused by mutation in the red-green photoreceptors which are found on the X chromosome. There is mutation which is on the red-green photoreceptors that are found on the X chromosome. They are the ones that are going to cause the red-green color blindness. Cycle between what is normal and colorblind C. We can see what the normal people see and those ones that are colorblind are going to see. So the normal people, this is what they are going to see. If you look at this structure here, there is a number in the middle. If you are not colorblind, you'll see that there is a number 45. Even here, there is a number in the middle. Can you locate that number? You can see it is 29. And here, there's also a number in the middle. If you locate it, you can see it is 6. So the normal people will be able to see those numbers. What of the colorblind person? What is he going to see? This is what a colorblind person is going to see. He sees nothing in the middle. He sees there is completely nothing there in the middle. So that is red-green color blindness. Normal people, that's what they are going to see. 45, 29, and then they will see the green. These are people who are not affected by the red-green color blindness. They are normal. Colorblind people, they don't see anything. They are seeing nothing. There is nothing they are seeing there. Nothing is seen. That is for a colorblind person. Genetic disease, hemophilia. Now, hemophilia is also an X-linked recessive condition. It is an X-linked recessive condition, and it is caused by mutation in coagulation factors responsible for clotting. Now let us look here. When you see here, there is everything here is okay. There is no mutation. You can see there is no mutation, no mutation, no mutation, no mutation. So the clotting factor here, there is no problem with the clotting factor. The reaction will go on if we have a damage here, and the vessel is going to be blocked here, and it will prevent blood coming out to go outside from inside, to prevent blood coming from inside going outside, because there has been a clot. That is the normal coagulation cascade. Now when you go to this one here, you are going to see this is normal, normal, the gene here is normal, normal. But when you reach this one here, you are seeing that this one here, there is mutation which has occurred there. Mutation has occurred there. And when mutation goes there, now this one becomes normal, normal, normal. Now when the reaction starts, the reaction starts and goes on and goes on. But when it reaches here, because here there has been mutation, the reaction will stop here. And if here we have been having a cut somewhere here, then clotting is not going to take place. And bleeding will continue. Blood will continue coming out. Bleeding is going to continue. Therefore, here now we say a person is hemophilic. A person is suffering from hemophilia and controlled bleeding. And this one we are saying it is found under X. It is an X-linked recessive condition, and it is caused by mutation in the coagulating factor. So this coagulating factor has been mutated, and therefore there is no clotting taking place, and that one is going to cause what we call uncontrolled bleeding. So hemophilia, we are saying it is an X-linked recessive condition. So the absence of the factor prevents clotting, and this results in a sustained bleeding. Because of this mutation we are having here, it is going to cause the absence of the factor that prevents bleeding. It is going to cause the absence of the factor that prevents bleeding. So this factor here that prevents bleeding is now made unavailable, and therefore there will be no clotting, and therefore we shall have a sustained bleeding or uncontrolled bleeding. Mutagens. Mutagens, these are agents that change or mutate the genetic material of an organism. A mutagen is an agent of change that changes or mutates the genetic material of an organism and causes mutation. We have different types of mutation. We have the physical, and under the physical we can have the ultraviolet rays, we have the X-rays. All those are physical. Then we have the chemical mutants. Chemical mutants like those ones that we find in cigarettes. We can find the chemical mutants in the processed foods. We can find them also in cleaning products. Those are chemical mutants. They are agents that can change the genetic material of an organism. Then we have those ones that are biological. Biological, these ones we can find them in viruses, and we can also find them in bacteria. So all those ones are mutants or mutagens. They are agents that can cause mutation or change in genetic material. A mutagen may be physical, it can be chemical, or it can be biological. Physical, chemical, biological, as we have already seen in origin. Mutagen may cause what we are calling genetic diseases, or they may trigger the formation of cancers. Those are the mutagens. They can trigger the formation of cancers. All those mutagens can trigger the formation of cancers, and they can also cause genetic disorder or genetic diseases. Hello, my friends. Thank you very much for participating. And today we have looked at Mendelian's genetics. We have looked at mechanism of inheritance. We have looked at the type of zygotes. We have looked at Mendel's inheritance. We have looked at the Punnett square. We have looked at the pedigree charts and the genetic diseases. Thank you. Please don't forget to subscribe.